Assalamualaikum all. Thank you for the opportunity to participate in the third International Conference of Science, Ethics and Civilization Concept 2024. Without further ado, I am Naim Aziz, representing the other co-author. Will share finding and information regarding our study on the effect of coastal and channel mangrove sediment on bioturbation distribution. First of all, mangrove are among the most productive marine ecosystem on Earth. They play an essential role in the ecosystem as natural barrier from destruction caused by ocean wave, tsunamis and storms. Necessary ground for fisheries, reducing coastal erosion, stabilizing shoreline, retaining nutrients and sediments, assisting in flood and river management, maintaining water quality. Supporting its sedimentary ecosystem, bioturbation activity is constantly ongoing, where organisms burrow into soil, reworking and modifying their environment. They play an important role in the carbon and nutrient cycle while shaping the soil geomorphology. However, differences in bioturbation activities on different physical properties in mangrove forests, as its coastal and channel have not yet been well addressed. Thus, this study observed coastal and channel sediment effects on bioturbation within the mangrove areas. The aim is to determine bioturbation distribution in coastal and channel regions by investigating the sediment characteristic and bioturbation frequencies along this region. To look at how coastal and channel mangrove affect bioturbation, the stratified random quadrat sampling method has been used for bioturbation frequencies, inhabited species, and population richness. The study has chosen Bagan Nahuda Oma, known as BNO, and Kilim Geoforest Park, known as KGP, as experimental sites to represent coastal and channel areas. For the mangrove species survey, we found that although some of the same species exist in both sites, but some different sites distribution were observed to differ between BNO and KGP due to differences in salinity and physical characteristic of the habitat. This is also due to the zonation, which refers to species tolerance and adaptation to certain environmental conditions. For sediment characteristics, sediment samples were taken using a corer and analyzed in the lab with the laser diffraction methods to measure particle sizes accurately. The analysis included statistical measures like mean, skewness, and kurtosis to understand environmental condition to its sediment characteristics. The finding indicates that sediment characteristics differ between KGP and BNO sampling areas. KGP sediment is poorly sorted, skewed toward larger particle sizes, and has a sharper peak in its distribution. In contrast, BNO sediment is better sorted, skewed towards smaller particle sizes, and has a less pronounced peak. This suggests that KGP is experiencing active sedimentation while BNO is less active. This is possibly due to lower energy levels or reduced sediment inputs. This finding is also supported by the different composition percentages in the two areas, highlighting the distinct sediment characteristic between coastal and channel mangrove. For the bioturbation frequency, the study counted burrow holes within the quadrats. However, it didn't delve into the detailed size and shape of the holes. The result demonstrates that BNO mangrove area has a constantly higher bioturbation frequency compared to KGP mangrove area. Bioturbation frequency also varies significantly across different zones, with the middle zone showing the highest values in both BNO and KGP sites. This variation indicates that bioturbation distribution is influenced by the specific characteristics of each mangrove area. In examining the effects of physical attribute influence on the bioturbation distribution, a two-way ANOVA assessment was carried out. The study has found that coastal and channel had different sediment distribution, and the bioturbation frequency were influenced by different inhabitants in both regions. Thus, the result of this study supports that coastal and channel region have significant differences in the bioturbation process. So, in a nutshell, bioturbation is a key ecological process influencing sediment structure, nutrient cycling, and microbial activities. The finding of the study suggested that the physical attribute of KGP and BNO do possess distinct characteristics that influence the difference in bioturbation frequency and organism biodiversity between coastal and channel mangrove area, thus reaching the study objective outlined earlier. Realizing the difference between the two types of mangrove from the point of view of bioturbation, this allows for better planning to ensure that human activity do not affect the natural biodiversity. The outcome of this study perhaps will increase awareness among the community members and their involvement in the enrichment and nurturing of ground areas, especially mangrove forests as highlighted in SDG 14, the life below water. Thank you for listening.